Hello and welcome. Uh, Today I am going to talk about another class of mutation we call back mutation or reversion. In my previous video on mutations, I tried to give you an idea about what mutations are and how these mutations are classified, taking different criteria. For example, one of the way of classifying mutations is the direction. Forward mutation, backward mutation. And I talked about forward mutations. Today, I am going to talk about backward mutations. Another way of classifying these mutations is in which type of cell they occur, germinal cells or the somatic cells. Another way of classifying mutations is at the DNA level. How many nucleotides undergo change? If it is single nucleotide change, we call it as point mutations. Two nucleotides involved, we call them as double mutations. Or there can be insertions, deletions, and the consequences of insertions and deletions. So coming back to today's topic, that is back mutation or reversion. So if you recall, I talked about what are forward mutations. That is, in a population, if a change takes place from wild type to a mutant type. For example, in a bacterial colony, which is able to synthesize proline amino acid, that's its phenotype. But when any one of the genes of proline biosynthetic pathway undergo mutation, it leads to change in phenotype. That is, the ability of cell to synthesize proline disappears and it becomes pro minus. So in a population of pro plus cells, when you see pro minus cells appearing, we call them as mutants or pro minus cells or pro minus mutants. That is forward mutation. Now, if in the population of pro minus cells, that is mutants, among the mutants, if you isolate this mutant, single mutant cell and multiply it, you will see large number of cells which are pro minus. So it becomes population of pro minus cells. So when in this population, when a cell undergoes a mutation, or you observe a cell which regains the wild type phenotype, that is the ability to synthesize proline, it is because of a mutation, or the original mutation or the forward mutation is being suppressed by another mutation. So that's how reversions takes place. What happens in reversion or back mutation? The first mutation is suppressed by another mutation. And we call that as reversion or back mutation. So what happens to conclude this part? That you have the regain of lost phenotype or resumption of lost phenotype to become wild type. So you have a wild type to mutant phenotype and from mutant to wild type phenotype. Okay. So this process we call reversion or back mutation. Okay, and um, and and the resultant cells, we call them as revertants. So reversion and revertants. Okay, now having understood this part, let us go about knowing how this back mutation or reversion takes place, or how the suppression of original mutations take place. So there are two ways. So there are two ways by which the reversion takes place. One is true reversion, and another is pseudo reversion. So what is this true reversion? In true reversion, the second mutation reverses the previous mutation, and the gene becomes absolutely wild type. For example, if in a gene sequence A changes to G, so in the first mutation, in the second mutation, G again becomes to T, to A. Okay, so it is the reversion of the original mutation to wild type. So the wild type sequence is regained completely, and so this is the true reversion. Okay, in pseudo reversion or false reversion, the original mutation stays, but the second mutation is such that it suppresses the original mutation. Okay, and the cell regains the wild type phenotype. Okay, so in pseudo reversion, what happens? You see the continuation of original mutation in the cell as it is, but the second mutation takes place, and that second mutation is such that it suppresses the previous mutation. And the second mutation can be in the same gene, so intergenic, or it can be in a different gene altogether. So second mutation can happen in the same gene, or in a different gene altogether. So, but the consequence is that it the cell resumes the wild type phenotype, and that is what we are going to look at in slightly more detail. But before that, another aspect of reversion is that reversions are the indicative of the kind of first mutation has happened. Means, as I told you, based on the changes taking place at the gene sequence level, it can be point mutation, it can be insertion, it can be deletion, and if it involves two nucleotides, it can be double mutation. So, by looking at reversion frequency, one can know that what kind of previous mutation was. Now, what is this reversion frequency one is referring to? Reversion frequency is the number of wild type cell obtained from total number of mutant cells. It is the proportion, basically, proportion or the number of cells converted into wild type cell from the total number of mutant cells. That is what we refer as reversion frequency or even mutation frequency. Mutation frequency is from wild type to mutant, number of mutants obtained from the total number of wild type cells. Similarly, here in the reversion frequency, you see the number of cells regaining wild type phenotype among the total number of mutant cells one takes. Okay, so if the reversion frequency is 10 to the power minus 8, which is the frequency for spontaneous mutation also. So that indicates that the original mutation was point mutation. So the reversion frequency, if it is 10 to the power minus 8, it means it involves only single change. But if the reversion frequency is 10 to the power minus 16, it means it is involving two nucleotides because it is the double. The reversion frequency is doubled. Why? Because two nucleotides are involved. So it is the indication for the original mutation. What was the original mutation? From where the reversion is taking place. So that can be determined by looking at the reversion frequency. Okay, so it can be point mutation, it can be double mutation, and if there is insertions, deletions of larger size of DNA sequences, these are there the reversion frequency is 0. Those kind of mutations cannot be reverted back. Okay, but if the reversion is single or double involving one or two nucleotides, then it is possible to have reversions. Okay, so this is another aspect of reversion, that by looking at reversion frequency, we can know what kind of mutation the previous cell has undergone. I mean, previous mutation was, whether it was point, double mutation, or insertion or deletion. If you are not able to get reversion, it means they are either because of insertion or deletion of large sequences. Okay. Now, let us try to understand the reversion process. When the second mutation is taking place, or the reversion is because of a second mutation in the same gene. That's what I told you. That second mutation can be in the same gene, intergenic, or the second mutation can be in a different gene altogether. And the second mutation is such which is suppressing the original mutation to give you a wild type phenotype. Okay, so let us first consider the second mutation in the same gene. So for example, if this is the wild type protein, this is the wild type protein, and this structure or functional structure is because of the presence of a positively charged amino acid here and a negatively charged amino acid here. Okay, so after mutation, what happens? This positively charged mutation, this amino acid is changing because of the mutation, and it is getting converted into negatively charged amino acid. And that is what happens in point mutations, that a single nucleotide change takes place, which changes the nature of, it changes the amino acid, not necessarily if it is not affecting the polarity of the amino acid, it results into silent mutation. Though the change is taking place, but the new amino acid is able to bring about the functions, so, but in this case, in this example, there is this amino acid is undergoing losing its polarity, for example, because of the mutation. So it becomes positive to negative. Now, because of this mutation, this interaction is not taking place, and this protein becomes inactive because it is not able to fold. Okay. Now, so you have the mutant cell, and when this cell multiplies, it will generate the mutant population. Now, if there is a reversion, that's what we are considering. Now, if there is a second mutation, now second mutation in one possibility is that the same nucleotide which underwent change is converted back to the original, the nucleotide sequence, and the the changed amino acid now becomes the original amino acid. Okay, that is the mistake which happens in the original mutation is corrected and corrected absolutely in the right manner. As I mentioned, if A nucleotide becomes G in a mutated cell, so in reversion, that same site, same place, same gene, G is becoming A, so that you get this protein again is able to fold itself and it becomes wild type. Okay, so this is true reversion. In true revision, this is what happens. Okay, now, now the another possibility is that the second mutation is happening same site, but the change is such that the changed amino acid it becomes plus to minus, but this minus becomes to plus. It may not be the same amino acid as in the previous wild type, but polarity of the of this same site mutation is happening, and the mutation is such that the mutated this minus becomes plus. So this is same site, second mutation in the same site, but it is not the wild type. Wild type. If for example, I'm just giving an example that glycine becomes lysine. So here lysine is again becoming to glycine, but here the lysine is not becoming glycine, but a similar positively charged amino acid. Okay, so this is second mutations, but not converting it to the original amino acid or original type of amino acid. Now, another way is, the third possibility is that this mutation stays as it is, but in the same gene, in this place, which was minus, it becomes positively charged because of a second mutation. So because of second mutation, this becomes negative to positive. And then when that happens, again the interaction is resumed. And you see a wild type cell. So if the consequences from pro plus to pro minus mutations here like this, then as a consequence of reversion, you see, a wild type. All are wild type reversions. Okay, all are wild type reversions. But here, if you see type 1 reversion, it is the true reversion because second mutation has reverted the mutated amino acid to the original wild type amino acid. Here, the nature of mutated amino acid is changed to the wild type. Now, if it was positively charged, became negatively charged as a consequence of the same site, same place, the second mutation, changing the polarity from negative, from positive to or negative to positive. Okay, but it is not true because the amino acid is different. Only the nature of amino acid is such that it is able to interact. But here it is again a second site mutation, but second site mutation is such that it regains the ability of protein to interact again because second site is here. This was negative in the wild type. It becomes positive so that the protein is able to interact and perform the function. Okay, so this is an example to illustrate what I was talking about. Okay, now, now this is very interesting. The second mutation in a second gene, entirely different gene, and that mutation, second mutation in second gene, is such that it suppresses the original mutation and gives you a wild type phenotype. This is very interesting. I repeat myself that Now we are considering the second possibility. That is a second mutation happening in another gene. And that second mutation is such that it suppresses the previous mutation to give you a wild type phenotype. Okay, so this is what we call intergenic suppression. Intergenic suppression. And also, this is one way of conditional mutations. Means the original mutation will show wild type phenotype in a particular genetic background. Okay, until if the second mutation is not there, or right kind of second mutation is not there, you will not see reversion. Okay, so it is in a way a conditional mutation. Okay, so there are many ways by which a second mutation suppresses the original mutation. Okay, the one and very important, and which I'm going to discuss or elaborate little bit in detail, is the informational suppressions. Okay, informational suppressions is the second mutation changes the way cell reads information on mRNA. Okay, that's very important. Okay, so second mutation is such which suppresses the informations. What kind of informations? It changes the way the cell reads the information of mRNA or on mRNA. That is one way of suppressing the original mutation by a second mutation in a different gene. Keep this in mind. I'll come back to this in a while. Now, interaction suppressions, overproduction suppressions, bypass suppressions, and physiological suppressions. They are very simple interactions because of a second mutation in a second gene. I'm not taking this part in this session. Maybe later I will address these things, but let us look at this informational suppressions, what these suppressions are, and how a second gene, which is involved in reading the information, which is located on mRNA, changes. You know, so in other words, what reads the information on mRNA? If you recall, it is a tRNA which reads the information written on mRNA. You know, that is during translation. It is the tRNA which recognizes a specific codon and brings a respective amino acid to that and keep adding. So it means the second gene which I'm referring to, or mutation in the second gene I'm referring to in this class of reversions, is the tRNA gene. Okay, so let us now, there are two types of informational suppressions. Okay, there are two types of informational suppressions. One is nonsense mutation. I have explained these nonsense mutation or chain termination mutations in my previous video. But here, what is nonsense mutation? Nonsense mutation is that a mutation in the gene, which results into the premature termination of the protein or the amino acid chain. Okay, so this is what happens in a nonsense mutation, that it results into the chain termination of the translated product. So you get, as a consequence, premature protein or half protein made, which is non-functional. Okay, and then you have missense mutation. Missense mutation again is the amino acid substitutions. Okay, the original amino acid, because of point mutation, is changed to something else, and that results into changed phenotype. Okay, So informational suppressions happen or there are two types: nonsense mutation, or we call them as chain termination, and missense mutation, call them as amino acid substitutions. Let us look at this cartoon. Okay, so if you see, this is the wild type mRNA. This is as an example, and it is making a wild type protein, and this is the wild type scenario. And now, if there is a mutation in this codon, that is UAC, becoming UAG, UAG, and the consequence is the UAG is one of the stop codon. Consequence is the production of premature protein, or half the size of protein, or truncated protein is the right word to be used. Okay, because of the mutation in this codon, which is getting converted from UAC to UAG. Okay, so this is the wild type scenario, this is the mutated scenario, and now how the reversions takes place. I told you three class of reversions, or three types of reversion. The first reversion is what we call true reversion, or class 1 reversion. That is, this UAG is precisely getting converted into UAC, that is the wild type sequence. So this UAG mutated version, this is forward mutation from wild type to mutant, and from mutant reverse mutation, back mutation or reversion is taking place because of the precise uh, change. So UAG is converted back to UAC because of mutation. Again, C is getting converted to G. Here G becomes C. True reversion. Okay, so this is class 1. Now, second is this is not getting converted into UAC, but it becomes UCG. Okay, UAG becoming UCG. It means this mutation stays as it is, but A is getting mutated to C. So original mutation is staying there. It's not changed. Okay, but this is converted to C. As a consequence, this codes for another amino acid we call serine. Okay, and this serine is doing functionally the same as the original amino acid, and you see the reversion here of the phenotype. This is also we call silent mutation. Though the second mutation is happening, second mutation and the first mutation also stays as it is, but the second mutation is such that it is suppressing the impact of the previous mutation. Why? Because the second amino acid, which is or the change or the mutation which is happening is such that it is changing the amino acid, and the nature of amino acid is such that it is able to regain the protein interaction to form the 3D structure, which is functional. Now, and the third type is, what is happening? The original mutation stays, okay? That is UAG is S. This is the mRNA, but there is a mutation in the second gene. That is a mutation in the second gene, and what that gene? That is the tRNA gene. tRNA gene for the lysine amino acid, which has the anticodon for this amino acid. Okay, so AAG, this is this is the mutation happening in the anticodon region of the tRNA, which is responsible for adding lysine. So that is becoming AAG, and that is able to respond to this UAG, which is the mutated, and it is able to then place then it reads this as a right codon for lysine and puts lysine there, So it is, again, to make you understand what's happening, the tRNA gene, the anticodon part of the tRNA, that undergoes, I mean, because that is also a gene or DNA that can undergo a mutation like any other part of the DNA. Okay, so that part undergoes the anticodon part of tRNA responsible for lysine undergoes a change. Therefore, that tRNA reads this mutated site as correct codon for lysine, and it adds lysine, and the chain is not terminated. The protein synthesis goes on, and it suppresses the original mutation. Original mutation is there. The only thing is, this is the mRNA. It is reading correctly and putting lysine there. Okay, and the complete protein is synthesized, which is functional. Now, okay, to make it more clear what is happening, this is the nonsense mutation happening. Here, this is the normal protein codon, for example, and this is the mRNA. Normal mRNA is produced. UUC and AAG. This is the DNA sequence. This is the mRNA sequence, and this is the protein. Complete protein is made. Okay, like this. After mutation, what is happening at the gene level? This TTC, out of TTC, one T is converted to A. It becomes ATC. Okay, ATC. So when it after transcription, you have this mRNA formed, which is having this mutated information. Okay, instead of AAG, you have UAG. UAG. Okay. Now this is the defective. If suppose in a mutant, the protein, the protein synthesis will be terminated at this point, because this is one of the stop codon. Okay, but then in a cell where the tRNA is altered, okay, this is what I was trying to say, that the anticodon of the mutant tRNA, so mutation is in tRNA, the second mutation is in tRNA, so this is, what is this change happening? It is, the mutation is in AUC, which is which codes for tyrosine, for example, and this mutated tRNA reads it correctly and puts tyrosine there, and therefore there is no premature termination, and you see the complete peptide being formed. Okay, so this is the example of second mutation in a second gene, suppressing the information, the way cell reads the message on mRNA. So see, the mutation continues, but because of the mutation in this tRNA, this is read as correctly, and the correct version is added, version of amino acid is added, and you don't see the premature termination. Now, missense amino acid or missense mutations or amino acid substitutions. Okay, like we saw in the example that if the mutation is happening from, say, positively charged amino acid, after mutation it becomes negatively charged. So from wild type to mutated version, and from mutant to revertant. So what is happening from polar to non-polar, and here non-polar to polar? Okay, so it is the substitution of amino acids, So reversion takes place because of the reversion of polarity of the amino acids. So how this happens? That is, there are three ways by which this can happen. That is, again, because of the second mutation in tRNA, because tRNA is the one which reads the information on mRNA. Okay, so I showed you one example in the previous slide, So, but then there are three ways by which it can substitute amino acids. One is that the mutant tRNA recognizes two codons. Two codons means one correct and another non-correct. Okay, or the mutant tRNA incorrectly recognized by amino acid synthetase and carries a wrong amino acid. So that is because of the mutation in, that is because of the mistake created by this enzyme, which activates the tRNA, So this enzyme incorrectly recognizes the tRNA and sends a wrong amino acid, or a mutant amino acid synthetase may charge an incorrect tRNA molecule. So that is because of the second, these two, because of the, because it involves the second mutation in amino acid synthetase, that is doing, that is not doing its job, which it is supposed to do, charging the right kind of tRNA to take the right kind of amino acid, okay, or charging a wrong mutated tRNA, which it should not, but it is doing that, and that's why that tRNA takes some amino acid and replaces the previous one. Now, these kind of suppressions are not fully effective in vivo scenarios, because this results into general substitution. Now, when something happens general, it is going to affect the cell growth. When a tRNA is mutated and it is replacing from everywhere the right kind of amino acid to a wrong amino acid, then the consequence would be the cell, either it will die and whatever cell survives, they will grow very, very slowly. Okay, so that's one thing. Now, another suppression of frame shift mutations. So if you recall, I told you that a single nucleotide addition or deletion can bring about change in the reading frame. Normally, it is the 3 codon, which are read. But if any mutation is because of addition of a single nucleotide, it means that codon will have 4 instead of 3. Or if it is deletion, the one nucleotide is missing, that will leave behind 2 amino acids, 2 nucleotides rather than 3. So when the reading, when the frame is read, it is going to change. That 4 means 3, 3, 3. So the reading frame beyond the mutated site is going to change, and the whole amino acid sequence is going to. But then what happens in the suppression of the original mutation of this kind? What happens that because of mutation in tRNA, it has, instead of 3, anticodon of 4. So it by mistake put something reading 4 nucleotide sequence for a codon, and then it is correctly identified. So because of mutation in tRNA anticodon region, same kind of mutation, and instead of 3, there are 4. Or deletion, there are 2 instead of 3. So it reads that particular mistake as correctly and adds something and move on, So this is the kind of informational suppression occurs of the original mutation, and brings about and it brings about the resumption of wild type phenotype. So to conclude, what I had been talking about is another class of mutation we call reversion or back mutation, which happens among mutants because of a second mutation, which results into resumption of wild type phenotype from pro minus cells to pro plus cells. Okay, and this happens two ways: true reversion, false reversion. True reversion is the correction of the original mistake or mutation taking place precisely, you know. So it is true reversion. The mistake or the change is completely reversed. Okay, and in pseudo reversions, the original mutation stays, but the second mutation is such that it tries to suppress the effect of the original mutation. And the second mutation, this kind of thing can happen in the same gene, intragenic or intragenic. There are various ways by which the second mutation in second gene can alter the phenotype from mutant to wild type. And I tried to explain to you what happens in very specifically informational suppressions, and how that happens. So I will stop here and request you to subscribe my channel and like this video so that you can get information about my future um, sessions. And in my next video, I'm going to address a very important question about mutations, that is, whether about spontaneous mutations, whether there's a random or they have any adaptive advantage. Very, very important question, which people asked, and of course it is answered for good, that whether the mutations have any adaptive advantage or not. Thank you so much.